Pascal, and yeah, thank you for the invitation. I'm a big fan of this seminar series, so it's fantastic to hear that you were open to give us the opportunity to present our recent work in that very exciting topic of language models for fitness prediction. So I'm part of Oatmeal, a department from the computer science department at Oxford, supervised by Yar and Gal, but we ended up doing a lot of collaborations with Deborah Mark's group at Harvard, and so the projects I'm going to be discussing today are very much a joint collaboration between our two groups over the past 2-3 years. And so to motivate the discussion, why should you care about fitness prediction in the first place? And just to give 3 reasons. The first one is that once you have a good model that connects a representation of a protein to its function, so once you have a mapping between a protein sequence, for instance, and one of its properties of interest, you can use that for many things. One, you can use that to do human variant annotation, so you can understand, for instance, the effect of genetic mutations and their propensity to trigger pathologies. You can use those models to understand the effects of mutations in viral evolution. So when a virus is mutating, in order for that virus to escape immunity, it needs to maintain fitness as a basic requirement. And so good fitness prediction allows you to understand that aspect a bit better. And more importantly for the talk today, they are extremely important to various protein engineering objectives. And so if we are looking at the sort of a prototypical process of protein engineering, we have 4 steps. Like after defining the requirements of the project and the property that we want to optimize, and the sequences that we want to start with, we typically go through the three steps. So in the next stage, we model those proteins. So we want to understand the mapping between their representation and their properties. There's an interplay between that model and a prioritization mechanism through which we are going to be able to, from a large pool of potential mutations that we could be interested in, which ones are the most desirable to go and test out in the lab. And then once we've selected that subset, we measure the properties of those sequences experimentally, and then we iterate. And a key ingredient of the process is this fitness prediction model. So this mapping between the representation of the protein and one or many of its properties. And so I'd like to present the broad typology of fitness prediction models. We can bucket them into 2 broad categories, and I will use that to structure the talk. The first one is the category of unsupervised models, which are increasingly becoming self-supervised models. And so we'll cover during the rest of the presentation family-specific, family-agnostic, and hybrid models. That will be the core of this talk. And towards the end of the presentation, we will also discuss supervised methods in both discriminative models and generative models, which is in which I will introduce a very recent architecture that we've been working on to do that. And so that will be the 5 steps of the presentation today. But I will first start with evaluation. How do we evaluate those models in silico? And so what we've been devising over the past year or so is this set of benchmarks called ProteinGym. It's a large collection of deep mutational scanning assays in which experimentalists have mapped the consequence of different mutations. And so they have measured experimentally the phenotype that is believed to be tied to the fitness of the protein, depending on the function of the protein. And once we have those experimental measurements, we can correlate the predictions of various models with those measures, and that allows to do like a very thorough comparisons of different models, depending on the type of protein. So depending on the depth of the multiple sequence alignment, for instance, depending on the taxon of those proteins, we try to cover like all possible taxa in these benchmarks. And we have both substitution assays, indel assays. We are increasingly adding new baselines to it. And everything can be accessed through our website. I'm going to put it at the bottom here. I will also make the slides available after this presentation. By the way, as I'm going through the presentation, I'm totally fine taking questions as we go along, or there will be also a natural breaking point in the middle of the talk, during which I can help address the first set of questions that come up. And so, very useful tool to benchmark those different fitness prediction models. If you have any suggestions about new models that we should add in, or new assays that we could also consider to further enhance this collection, feel free to reach out to us, and we'll be happy to collaborate with you on this. So without all that said, so now let's deep dive into the different methods for fitness prediction, and I'm going to start with the family-specific prediction models. And so, remember, we are in the realm of unsupervised fitness prediction models. So we are going to try and perform this mapping between typically a sequence, a protein sequence, and the fitness of that protein sequence, without relying on any experimental label at all. And the way, like one of the most popular way to do this is to extract a set of homologous sequences from a particular protein of interest that are all evolutionarily related. So you typically obtain this through a multiple sequence alignment. And then once you have this multiple sequence alignment, you're going to try to learn a distribution over the sequences in that MSA. And historically, it's been very models looking at initially single residues, so site-independent models, or pairs of residues with POTS models like the mutation, and more recently, full generative models of the full sequence, such as deep sequence, EVE, and GENA. And these models are very powerful. They've obtained very strong performance, but they have a few different limitations. One limitation is that because they are trained on a multiple sequence alignment that acts as sort of a fixed coordinate system, they are unable to score insertions and deletions. And indels can be very important in lots of practical applications. So that's one key limitation, like that you can use them to score indels. Another one is that for some protein families, it's actually difficult to get a very deep multiple sequence alignment. And so the performance of those models is typically related to the depth of that alignment. And so there's a limitation in terms of the scope of different proteins that you can go and model with those types of approaches. And lastly, and this is more something that is dissatisfying if you were to train those models, for instance, in the EVE project, which is featured in the center of this page, we ended up looking at thousands of different proteins, human proteins, and at each time, for each new protein, we had to restart the training process from scratch. And so we ended up training thousands of different TAEs that share nothing in common. They're always retrained from scratch. And so that feels like a bit dissatisfying. Intuitively, there should be some information that we could share across protein family. And that's the intuition behind what we call family-agnostic models, so models that are trained across protein families. And the first of such models was UniRep, which was an LSTM model trained across a wide range of different protein sequences from the UniRep database. And subsequently, with the advent of transformers, there's been a lot of improvements on top of UniRep. One of the very popular one being ESN1V by the team at Meta, that showed that you could get also very good performance with those models with a few tricks that I will describe soon. But like the process to do that is you train a large transformer model across across a large portfolio of sequences, and you learn patterns that generalize across protein families. There are still a few limitations with that process. The first one being that with that masked marginal approach that is used, for instance, in the ESN1V paper, you cannot use insertions and deletions as well. They are that heuristic also introduces an approximation such that the dependencies between masked position, when you look at multiple mutants, for instance, is not taken into account. And so when you evaluate the effect of multiple mutants, you might be ignoring potentially important epistatic effects. And lastly, the performance in a purely zero-shot setting of those models is typically lower than for family-agnostic models. And so what the team at Facebook did to match the performance of deep sequence back when the paper was published was actually to first train an ensemble of 5 large transformer models, but also fine-tuning those models on a set of sequences that are related to the sequence that you want to score. So you fine-tune those large models on MSAs, and with that process, you can match the performance. And so that led to this new breed of models that combine properties of family-specific models and properties of family-agnostic models called hybrid models. Now, different ways to hybridize those models. So the first one is what I was just referencing with respect to the spiked fine-tuning in the ESN1V paper, or also that idea was very much present in the original UniRep paper with the notion of Evo tuning, which was the same idea of training the language model on MSAs. Another way to do this is, instead of fine-tuning the entire coefficients of the model, you can fine-tune a prompt. And so that's an idea that we tested in a paper called Rita. And so like you have a soft prompt that you fine-tune on a set of related sequences to the family that you're interested in, and then subsequently, when you want to score the sequences in that family, you condition with that family-specific prompt. Another way to hybridize the two types of models is achieved through something that was introduced in the MSA transformer, where you're going to learn a model of multiple sequence alignment across a wide range of different protein families. And this is a very elegant concept that allows to get very good performance for contact prediction, but also for fitness prediction. And the last one, which is what I will be focusing a bit more prominently in the rest of this presentation, is to do retrieval. That is, you still have as your backbone family-agnostic language models that you instill information from homologs at inference time. And I will be a bit more specific in the next few minutes. So we'll be focusing on that in the next few slides. So this idea of retrieval at inference was introduced in a paper called Tranception. And Tranception has 3 key ingredients. The first ingredient is, and I will cover those 3 ingredients in what comes next. So we have the Tranception inference, we have the retrieval inference, and then we bring them together to get state-of-the-art performance. So in terms of the Tranception inference, which is like this first key ingredient, there are 2 aspects of it. The first one is what we call Tranception attention. So the motivation for Tranception attention was we wanted to leverage autoregressive transformers, and that was primarily to get around the limitation that I was talking about with respect to masked language models and the fact that they were ignoring the epistatic effect between the mask position when you focus on multiple mutants. And autoregressive transformers are not subject to those types of limitations because they are generative models of the full sequence. And so you can you don't have to make any approximation like the masked marginal approximation I was discussing earlier. You can get directly the likelihood of the full sequence, including like all of the multiple mutants in that sequence in one go. And so that was one of the initial idea, was to leverage autoregressive transformers. And on top of the standard GPT-like architecture, we also wanted to promote 2 things. So the first one was to more explicitly extract patterns at different resolutions. So different, like to instill this notion of k-mers into the architecture. And the other design principle that we wanted to convey is to have head specialization. So different attention heads focusing on different resolutions. And that led to this concept of Tranception attention, where we start from the standard autoregressive, so the standard transformer decoder architecture. But then we apply a convolution for each. We split the attention heads into 4 groups, where each attention group has a convolutional kernel of different length that is associated to it. And so, for example, in that second group here, we're going to apply convolution of kernel size 3. We apply this convolution right after the linear projection to create the key, the queries and the values. So that allows to more explicitly mix the information at that level of resolution within that group of attention heads. And we replicate the process in parallel across all of the different attention heads. So that's the first ingredient. The second ingredient is what's called grouped alibi. So we built on top of alibi, which was a concept that was introduced in NLP. It's a different type of positional embedding, where instead of adding position embedding at the very beginning in the representation of your tokens, you're going to instead penalize the attention matrices with coefficients that depend on the distance between amino acids in the sequence. And for things to be working well with our scheme, with our head specialization scheme, we're also performing this scheme, learning the different coefficients of different resolutions across these different groups. So we're sort of applying an alibi for each group of attention head is the better way to put it. And that is working very well. It's something that you should consider if you're interested in applying transformer even to other types of problems. So we've looked at protein sequences here, but that's an architecture that is agnostic to the type of data modality that you're interested in, and that might be important if you're focusing on RNA models or DNA models or molecules or something else. And that works very well in practice, both in terms of the validation loss compared to, we did this ablation compared to the standard GPT architecture. Primer, which was the paper that first introduced this concept of convolving after the linear projection to create key values in place. So both better validation perplexities, but also, and more importantly, better downstream task performance. So there's all of that described in the paper, but you should consider that mechanism in your project eventually. That's a very we found it to be a very powerful transformer architecture. So that was the first ingredient. The second ingredient is this notion of retrieval and inference. And very simply, what we are doing is, say if we want to score the fitness of a given input sequence, we're going to retrieve a multiple sequence alignment for this particular sequence. And then in a way that is very analogous to what the way things are done in a site-independent model, we're going to extract pseudo-counts of the amino acids that we find at each positions, at each residue in the multiple sequence alignment. And it will give us another distribution of the amino acid vocabulary at that position. And that's very powerful then once we combine that mode of inference with the mode of inference from the autoregressive transformer, right? So in blue, we have this, if we want to make a prediction about this token here in the middle of the sequence, we can use the autoregressive transformer to make a prediction of this position condition on the left context, but also based on the empirical distribution that we see of amino acids in the corresponding multiple sequence alignments. And those two are like very like two complementary ways to look at the same problem, and it works very well in practice. Like they combine very nicely together. There's a slight nuance that we also do in the paper, is that instead of just relying on the left context when we make a prediction of a particular token, we also do the reverse. We look at we apply the transformer from right to left to symmetrize that process, and then we average the prediction at each position, so that we take into consideration the two contexts. And to be able to, for that to be working well at Inference-time, we apply during training a data augmentation. So we symmetrized, we flip the sequences randomly during training, and that allows the model to learn from the two directions. And so that works very well in practice. We get an architecture that is working very well. So like the top table is looking at the performance on the ProteinGym benchmarks that I was talking about earlier. So this is specifically the superposition benchmark, and looking at those 90+ DMS assays, but grouped by the depth of their MSA. So based on the low MSA group, medium depth of the MSA, and high depth. And we see that Tranception with that with the retrieval at Inference-time is obtaining very strong performance in the low MSA depth regime. And that's exactly what we designed it for. We wanted to go after those proteins that are potentially a low depth MSA that would not be well covered by the family-agnostic type model like EVE and other alternatives. And not only it performs well in the low depth regime, but what we saw is that we run an analysis in which we took all of the different multiple sequence alignments, and then we progressively censored some of the sequences from those alignments based on the similarity to the wild type in the MSA. Typically, when you do this, so at 100%, when you keep all of the sequences, you're on the left of this chart. The performance is very high for all of the different models. But as you start to progressively remove sequences that are the most dissimilar to the wild type sequence, you see that the other two types of models that we compare against, so in this case, EVE and MSA transformer, were less robust to the depth of the MSA. And so the performance was dropping quite rapidly as you were working with shallower alignments, which was also something that was interesting to see from a practical standpoint. You want your method to potentially be a bit robust to the characteristics of the MSA, that the MSA algorithm that you use to get that alignment, and also, I mean, it goes back to the point of having a model that is more robust to low depth proteins. Another advantage of this architecture, and in particular the fact that the inference is the retrieval is done at inference, is that it provides a lot of flexibility, because this model, this MSA that we are retrieving, if we do have, for instance, particular domain knowledge for a protein, so we give the example here of the BRCA1 protein, where there's like two very clearly identified domains in that protein, and we can get higher quality alignments of each domain if we treat them separately. And so if you were to model this protein, factoring in this information and retrieving your MSAs with that in mind, you can get even higher performance in terms of fitness prediction by retrieving those two domains separately and then combining with the autoregressive transformer. And another advantage is that we can actually leverage a model that is a bit more complex than just counting amino acids in the MSA to generate synthetic distribution of residues of amino acids at that residue. We can, for instance, leverage those more complex family-specific models of the alignment, and that's an analysis that we did in the appendices of that Tranception paper that we wanted to explore a bit more in a subsequent work called TranceptEVE, which is exactly this idea of still combining a family-agnostic transformer model. So we still keep Tranception, but instead of using this very simple site-independent model of the MSA, we leverage an EVE model, and this is working even better in terms of fitness prediction performance, with of course the trade-off that it takes a bit more time to compute to train the EVE model that is used to make those predictions. And so let me share a bit more information about this model. So yeah, so as we're saying, two components. The Tranception component is exactly the same as what I was describing earlier for the original Tranception model. The EVE inference is the different parts, and what we do is we retrieve a multiple sequence alignment for that family. We train an EVE model on that MSA, and instead of just ensembling those two models, it's not as if we were predicting things with Tranception and predicting things with EVE and then averaging the two predictions. This is not what we do. what we do is that we train that Eve model to learn this static log prior of the protein family. And so more specifically, so Eve is a underlying Eve, and one of the key components of Eve is this Bayesian variational autoencoder, where we are training on this MSA, and we learn to encode and decode sequences with that architecture. And from the latent space to the output, the decoder provides, or rather, the last layer of the decoder provides a log probability of the amino acids in the vocabulary at each position. But like those probabilities, they factor in the dependencies between all of the positions in the sequence, because they depend on the latent representation that characterizes the entire sequence, and that gives a very good log prior that you can ensemble, or that you can average with the Tranception transformer, the same way that we were averaging the Tranception autoregressive prediction with the residue specific predictions. So that's one of the key concepts underlying this architecture. The second idea that contributed to the performance is the parameter that averages the two types of the two modes of inference between Tranception and Eve depends on the depth of the multiple sequence alignment. And so at a high level, you have a model that sort of adapts gracefully to the depth of the multiple sequence alignment. If the multiple sequence alignment is very shallow, this averaging coefficient is going to be very much in favor of the autoregressive predictions. So like your predictions are going to be mostly based on what the autoregressive transformer thinks. And if conversely the multiple sequence alignment is quite deep, then the model will lean more heavily on the Eve predictions. And that works very well in practice. And when we look at things on the ProteinGym benchmark, we are getting strong performance across the board, both in terms of the depth of the MSA, in terms of the mutational depth. So looking at singles, doubles, triples, etc. Looking at different taxa, and also looking at indels. So that illustrates the power of combining strong models that are learning dependencies across protein families, and models that learn very complex distribution over the residues within a given family. And so this is sort of a natural breaking point here of this presentation. I'm going to pause here and see if there's any quick question in the chat. If there's none, I can continue, and I'll keep time at the end for more questions. So let me see. I don't see any question yet in the chat. Feel free to post it. I'll wait a few seconds. If there's nothing at this stage, I'll just continue in the interest of time. Okay, so okay, so there's a question from Pradeep in the chat. Could you describe the random mirror sequence augmentation in more detail? Yes. So yeah, so it's very simple. It's when you're forming your mini batch when training the architecture, you're going to randomly flip the sequence in some of the sequences in your mini batch, so that what you want your model to be able to learn is to if there's, for instance, some dependencies that would differ, looking at sequences from left to right or from right to left, you want to be your model to be able to have been trained on capturing those dependencies, and to be able to do that, we simply flip some of those sequences at random during training time, so that at inference time, when we do this scoring from both sides, the model has been trained to handle sequences in those two types of situations. And so it's able to get good performance in that setting at inference time. And continue. Oh, and I didn't cover this. That's another analysis that we carried out in the TranceptEVE paper, which was to also look at the ability of those models to predict the effect of mutations in humans, in comparing, instead of so there are two axes on this plot. The x-axis, the y-axis is the Spearman with the experimental measurements, so pretty much the ProteinGym performance that I was describing earlier. The x-axis is the AUC in terms of the actual clinical labels, pathogenic and benign, that are assigned by clinical geneticists, for instance, for different genetic mutations. And there, the model was able to perform very well on those two types of benchmarks. So that's for the realm of unsupervised models for fitness prediction. Now I'm going to cover 3 different types of models: supervised models, but as we will see, I just presented in the first part of the talk, is actually going to be helpful to improve the performance of those models. So let's dive in. So supervised fitness predictors discriminate between two different kind of models: discriminative models versus generative models. And so the discriminative models, they are sort of the standard way that things have been done in the field, where you typically combine 2 ingredients. The first ingredient is a representation of the proteins, and so it can be one hot encoding of the amino acid sequences. It can be physical chemical property features that you extract for the proteins that you're interested in, or it could be recently what has been a popular approach is to extract the embeddings of those proteins with one of the models that I was describing, whether it's Eve or it's N1V or the NSA transformer. And once you have a representation of those proteins, you plug that into a simple regressor, ridge regression, random forest, etc., that you then train on the labels that correspond to the property that you want to optimize. It's sort of a standard process that has proved very powerful in practice. And so, like in that realm of models, there's large language models can be used to get embeddings. Another way to use those fitness prediction models is to augment the prediction from those models with the unsupervised prediction. I mean, that's a method that was described in this paper by Chloe Su. That was also presented in this seminar series maybe a year ago, maybe a bit earlier than that. And that's working beautifully well in practice. And so you sort of have your baseline prediction that comes from an unsupervised model, and you combine that with one encoded features for that protein sequence that you want to score. You plug that into I think it was like a ridge regression in the case of that paper, and that gives very good performance. So that's the realm of discriminative models, and they work very well in practice. Recently, we've been working on this new architecture. So like it's brand new work. The preprint is not out yet, but I just described the key principle behind this idea, and it's quite powerful. This method is called ProteinNPT, so NPT for non-parametric transformer. It falls into the realm, the broader realm of generative supervised fitness predictors, and you can think about it as sort of a hybrid between two existing models. So you can think about it as a hybrid between the MSA transformer and the non-parametric transformer, which is work from colleagues at Oatmeal from the same group at Oxford that focuses on modeling on predicting properties in a tabular data setting. And so like if you're interested in that line of work, these are like the two main papers to read to understand this the concepts I'm going to be talking about. The idea is that instead of learning a representation of a single sequence at a time, you instead passes the input to your model is a group of sequences. It's not a single sequence, it's a group of sequences. And you're going to, so in this case, we are looking at a variant of actual transformer, like in the MSA transformer paper. We are going to combine both cross-feature attention. So in this diagram here, in gray, like the embeddings for the different amino acid for a protein sequence family I'm interested in, and like the different rows are different mutated sequences that I'm interested in predicting. In this blue shade, it's the embedding of the target that I'm interested in predicting for those different sequences. And I'm going to pass in as a single instance to the model this entire data set, like all of the sequences at once. And the model is going to learn these dependencies across columns and across rows. Like you apply the two types of attention, similar to the way it's done in the MSA transformer, except that here you have also a representation of the label that you're interested in predicting. And depending on what you want to predict, it's very helpful to instill that information, the information from the experimental labels that you've spent a lot of time and resources to collect to further enhance your model. And so you train your network with this actual transformer architecture. To the difference of the MSA transformer, we now have two types of losses. So we had the same loss on the same cross entropy loss for the masked token prediction task, the same way that there is in the MSA transformer. But we have a second component to the loss that corresponds to the reconstruction error from predicting the actual target value. We train the entire architecture with a loss that combines those two objectives. And there are like many advantages to this. The first one is that you actually get very compelling performance. And one of the reasons why you get strong performance, at least my interpretation of it, is that if we go back to those models, right? When I was saying, we take the embeddings of the protein sequences and we use that to plug into a simple regressor. Typically, what is done in practice is you're not going to fit the embeddings each amino acid sequences for all of the tokens in the sequence, because that would be too high dimensional of an input. You sort of need to apply some method for dimensionality reduction, and it could be PCA, or it could be simply what people have done in practice is just to take the mean embedding across the different tokens in the sequence, and that has led to good performance in the past. But intuitively, when you do this type of mean pooling, you lose potentially a lot of very valuable information in the process. And here we're not doing this, and that allows the architecture to apply attention to the particular positions in the sequence that are critical to predict the target that you're interested in, and you don't, smudge everything together into a single representation of the inputs and plug them into your regressor. And that's probably why it's working nicely. The second benefit is that it easily adapts to multiple objective prediction. You simply need to add as many target columns as you have objectives that you want to optimize across. And that's the added. Not only it's easy to modify the architecture to handle this scenario, but also you're going to be able to quite easily capture the dependencies between some of those properties. So, for instance, you might have partial labels for some sequences, but not for others. And with that, you're going to be able to, when you make a given prediction, you're going to be able to capture those dependencies very nicely with the same attention mechanism. And the last benefit of this architecture, and that is most relevant for this seminar series, is that you can then use this construct to condition the generation of new sequences based on certain property values. So I could, for instance, condition on high values of the properties and have the model architecture generate new sequences that is conditioned on that high value of the property. And I can also put examples of candidate sequences that I have access to, and use these to also condition the generation of tokens, and sort of that can be a useful tool for protein design. And I see that Umberto is in the chat. That's very much that would be very much inspired from work that has been done by him and Damiano Scarbosa, where they were generating sequences with NSA transformer. And so I'll link the paper in the slides as well. But instead of doing this fully unsupervised, you could do pretty much the same process, but conditioning on certain property values of interest. And that could be interesting. The limitation, of course, is that it requires a higher computational budget, both in terms of time to train the model, but also in terms of memory requirements. But we use the same type of tricks to make things more tractable as what is done in the NSA transformer. That is, we do in the architecture successive row attention layers with column attention layers, or the reverse. And we also, in terms of memory complexity, we do this tied row attention trick. Is the relationship that we want to learn is for the same reasons that are put forward in the NSA transformer. This relationship across rows are consistent, and so that we use that same tied row attention trick. And I will conclude this presentation by thanking all of my wonderful collaborators on these various projects. I talked about even Tranception, talked about TranceptEVE, and the ProteinNPT project is something that Ruben and I are currently working on. We hope to have the preprint out in the next few weeks. So stay tuned for that. And of course, thank you very much for my supervisor Lauren and Debbie for leading the charge on several of these projects. I'll take the next few questions in the chat. So there's a question from Marsha. Would these models translate easily to multiple objective property prediction, affinity versus stability, for example? And Marshall, I'm not sure if you were referencing to, I'm not sure which types of models you are referencing. There's the ProteinNPT. I think I addressed this question afterwards. It's very easy to do multiple objective optimization. So you would have in your example, you have one column for affinity and one column for stability, and you can do things like this. For other models, you would, for instance, for the discriminative type model, I think you would simply have a different prediction head for different objectives, and then you would have to rely on sort of the most standard approaches to do that. Question from Evik. Yes, I will make the slides available after the presentation.
All right. Any last questions before we wrap up here this afternoon? All right. Well, thanks again, Pascal, for that awesome presentation. As usual, our recordings will be available on YouTube, and Pascal can share his slides, on the Slack channel, or you can find them on YouTube as well, which is a little more indirect. But in any case, be sure to check out our links for future speakers and join our email list and join our Slack channel if you haven't already. And yeah, thanks again for joining us, Pascal.
Thanks again for the invitation, and thank you all for the questions.